Discovery of cell. Robert Hooke, a British biologist, in 1965 examined a slice cork under his self-designed microscope. He saw that the cork resembled the structure of a honeycomb consisting of many little compartments. Robert Hooke called these boxes as cells. In Latin, cell means a little room. This was the very first time that Robert Hooke observed that living things appear to consist of separate units. Structure of different types of cell. Both plants and animals are made up of cells. These organisms have many cells grouped together in a single body. In human beings, different body parts are made up of different kinds of cells such as blood cells, nerve cells, bone cell and smooth muscle cells, etc. It means different body parts of the human body are made up of different types of cells and perform different functions. Let us discuss the cell structure of some simple organisms. Amoeba is an organism generally found in the salt or freshwater bodies. It is made up of a single cell, thus it is called as unicellular organism. There are no different body parts in the unicellular organism. Microscopic studies of the cell conclude that each cell has three features: plasma membrane, nucleus and cytoplasm. Cell. Take an onion and cut it into two parts and then, with the help of forceps, peel off from the concave side of the onion. Keep it in the watch glass containing water. Take a glass slide, put a drop of water on it. With the help of paintbrush, transfer a small piece of the peel from the watch glass to the slide. Now we put a drop of iodine solution on the slide. Put a cover slip with the help of dissecting needle on the slide. Observe the slide under a compound microscope. Honeycomb-like structures can easily be observed. Precaution taken during this experiment. The peel is perfectly flat on the slide. Avoid air bubbles while putting the cover slip. Structural organization of cell. Plasma membrane. Plasma membrane is the outermost covering of the cell. It separates the contents of the cell from its external environment. Plasma membrane allows or permits the entry of some materials into the cell, such as oxygen. It also allows exit of some materials such as carbon dioxide out of the cell. During cellular respiration, carbon dioxide accumulates in high concentrations inside the cell. In the external environment, the concentration of carbon dioxide is low as compared to that inside the cell. This difference in concentration influences carbon dioxide in the cell to move out of the cell. Similarly, concentration of oxygen inside the cell becomes low in comparison to its external environment. The movement of water across the plasma membrane is due to osmosis. If the medium surrounding the cell has a higher water concentration than the cell or is a hypotonic solution, water enters into the cell by endosmosis. If the medium has exactly the same water concentration as the cell, or in other words is isotonic solution, there will be no movement of water across the cell membrane. If the medium has a lower concentration of water than the cell or is hypotonic solution, the cell will lose water by osmosis. Cell organelles. Endoplasmic reticulum and Golgi apparatus. Every cell has subcellular elements called cell organelles, which play a crucial role in cellular activities. Structurally, endoplasmic reticulum are long tubules or round or vesicles in shape. There are two types of endoplasmic reticulum: rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. RER looks rough as ribosomes attached to its surface. Ribosomes are the sites of protein manufacture. The SER helps in the manufacture of fat molecules or lipids. Endoplasmic reticulum serve as channels for the transport of materials between various regions of the cytoplasm or between the cytoplasm and the nucleus. The Golgi apparatus was first described by Camillo Golgi. In Golgi apparatus, vesicles arranged approximately parallel to each other in stacks are called cisterns. Material synthesized near the SCR is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. It helps in the storage, modification, and packaging of the products in the vesicles. Structural organization of cell. Cell wall. In plant cells, there is another covering lying outside the plasma membrane called the cell wall. The plant cell wall is mainly composed of cellulose. Cellulose is a complex substance. The important function is providing strength to the cell. An important function of cell wall is to maintain turgor pressure. Cell wall functions as semi-permeable membrane. It also acts as first line of defense for the cell during an attack from pathogens and microorganisms. Structural organization of cell. Nucleus. The nucleus has a double-layered covering called nuclear membrane. Cells having a nuclear membrane are called eukaryotes. The nuclear membrane has pores which allow the transfer of material from inside the nucleus to the cytoplasm. The nucleus contains chromosomes, which are visible as rod-shaped only when the cell is about to divide. The chromosomes contain information for inheritance of features from parents to next generation in the form of DNA molecules or deoxyribonucleic acid molecules. The functional segments of DNA are called the genes. In some organisms like bacteria, the nuclear region of the cell may be poorly defined due to the absence of a nuclear membrane. Such an undefined nuclear region containing only nucleic acids is called as nucleoid. These cells are called prokaryotes. Let us tabulate few differences between prokaryotic and eukaryotic cell. Cell organelles. Lysosome and mitochondria. Lysosomes are a kind of waste disposal system of the cell. Lysosomes have powerful digestive enzymes. It engulfs waste material and breaks down into small pieces. Lysosomes are also known as the suicide bags of a cell. Mitochondria are known as the powerhouses of the cell. Mitochondria have two membrane coverings. The outer membrane is very porous, the inner membrane is deeply folded. Energy is released by mitochondria in the form of ATP or adenosine triphosphate molecules. ATP is known as the energy currency of the cell. Cell organelles. Vacuole and plastids. Vacuoles are small size in animal cells, while plant cells have very large vacuoles. Vacuoles are the storage sacs for solid or liquid contents. Substances such as amino acids, sugars, various organic acids, and some proteins are stored in vacuoles. In amoeba, the food vacuole contains the food items that the amoeba has consumed. Plastids are present only in the plant cells. Color plastids are called as chromoplasts, whereas colorless plastids are called leucoplasts. Plastids containing the pigment chlorophyll are known as chloroplasts. The internal organization of the plastids consists of membrane layers and stroma. Chloroplasts are important for photosynthesis in the plants. Cell organelles. Endoplasmic reticulum and Golgi apparatus. Every cell has subcellular elements called cell organelles, which play a crucial role in cellular activities. Structurally, endoplasmic reticulum are long tubules or round or vesicles in shape. There are two types of endoplasmic reticulum: rough endoplasmic reticulum RER and smooth endoplasmic reticulum SER. RER looks rough as ribosomes attached to its surface. Ribosomes are the sites of protein manufacture. The SER helps in the manufacture of fat molecules or lipids. Endoplasmic reticulum serve as channels for the transport of materials between various regions of the cytoplasm or between the cytoplasm and the nucleus. The Golgi apparatus was first described by Camillo Golgi. In Golgi apparatus, vesicles arranged approximately parallel to each other in stacks are called cisterns. Material synthesized near the SCR is packaged and dispatched to various targets inside and outside the cell through the Golgi apparatus. It helps in the storage, modification and packaging of the products in the vesicles.